We can't hear you if you're speaking, sorry.
Oh, I'm so sorry. I'm so sorry. I was in mute all this time. Sorry about that. So, you know, I think now you heard me, right?
Yes, we could hear you.
Okay, okay. Sorry about that. So, mainly next 16 weeks, what we are going to learn from this course. Um, what different type of microscopes is available and what these each microscopes application and functions and how to process the samples and how to interpret the images and how you can correlate different type of microscopes images along with clinical parameters. So we will discuss all these with each and every microscopes. So the first thing is the aim of the course is give you a good understanding of the theoretical basis of microscopy. As you know, most of you guys, when you are in the high school or undergrad, even now, masters, if you are working in the lab, you will always, opportunity to use the microscopes. But how do you know that, the microscopes works? Because in the lab, we know we have microscopes, seniors or your mentors or research professors will teach you, okay, this is a slide and you can see under the microscope, you see the images and take a picture and you interpret. But if you wanted to improve the quality of your image and quality of your samples and, the interpretation, it is all these three is very, very important to validate your, data. So for that, you need to know that the good understanding of the theoretical basis of different type of microscopes. Also, get the images, and most information from your samples. So that's what, when your goal is not only just to process samples or not only just to you observe under the microscope, but you have to get ultimately the best images. Then only you will get a detailed structure of any samples. So that is another important aim of this course. Also, how do we process? So, the process is very, very important for any microscopes. Because if the process is not, properly handled, then your images will be affected. Ultimately, your, the informations which you get from this is not valid. So we have to make sure that, how important the microscopes are, how to handle the microscope, what is the information so you need to know, how the functions are, how we can improve the quality of the images and how we can interpret the images, the structures. So that's we are going to, see in next 16 weeks. Also, this course is which gives you an opportunity to understand the basics of, the structure of any your given samples which you observed under different type of microscopes. Also, you should know once you see the image, whether it is comes from light microscopy or electron microscopy or confocal or anything, any different microscope. How do you know that this image is took from that particular microscope? Is you need to know all these details. You have to study all the methods. You have to observe the samples from different microscopes. So then you will familiar with what kind of images you are going to get from these different microscopes. That will gives you, the important informations. Understand the operation and function of a light microscope, fluorescence microscope, and electron microscope. So this is a basics. We are not going to teach you the physics of complete the microscopes, but he will give you a basic ideas about how these microscopes works. Also, preparation. So, what's the goal of your preparation? What are all the factors which affects the quality of your, samples? That is, you are getting images from this process of samples. So these are all very, very important and learning goals of this, in this course. So how we are going to, achieve these kind of informations? So your basics, like, there are 16 weeks divided into, 16 weeks. So it is not in particular order, but I give you some basics of how we are going to, go through next 15 weeks. So our goal is like, basically we are going to study about a different type of microscope. One is light microscope. The other one is, technique is the fluorescence microscope. Another technique is laser capture micro dissection techniques. The other one is a scanning electron microscope, transmission electron microscope. So we are going to see all this, in this class. So how the, basics, theory and practice of light microscope, and also how to, operate, how you can improve the, quality of your microscope so that you will get a detailed structure. So that is our first few weeks. We are going to discuss about the light microscopy. Also, like a fluorescence microscopy. That is, how important this fluorescence microscopy and why we wanted to use the fluorescence microscopy, even though already we discussed about the light microscopy and which gives you a structure, and what else you are going to get it from the fluorescence microscopy. And that we will discuss in the following weeks. So then, we are going to talk about more on, electron microscope. So, like a light microscope and electron microscope. Why we called it is light microscope and this one is, electron microscope. So in electron microscope, very important for biological, science. One is a scanning electron microscope. The other one is transmission electron microscope. So the scanning electron microscope is again, the basics of electron microscope. What's the mode of operation and what type of information you are going to get it from this, scanning electron microscope, and also what is the limitation, what is the advantage, what kind of information you are getting from this scanning electron microscope, and how you will, store these images, and whether you are going to get only the 3D images or else you can get it from the scanning electron microscope. So that we will discuss in weeks 5 and 6. Then the transmission electron microscope is important in electron microscope, and which is very expensive, and we are going to get what kind of images we are getting from this and how process the samples. These will be discussed. So laser capture micro dissection is another important technique that we are going to discuss about this, and what is the laser capture micro dissection and how this is differ from other light microscope like a bright field microscope or a fluorescence microscope. And this we will discuss that also. And also whether this technique is only, are we going to observe the structure, or we are going to do something else with this technique. So we will deal that with discuss in the coming weeks. So the last week of this course, that is April 23rd. So that is only one written test. There is no like a midterm or something, only one final test. Whatever you learn from these 15 weeks, whatever material which I provided is enough for the exam. Okay, so you no need to go for any references or books or etc. So whatever the material which provided that is sufficient for your exam, and also the PowerPoint which we discussed here the lecture. This is very important for your exam. So the last week, the 16th week, that is April 23rd. That is your final exam. That would be maybe in person or WebEx with exam soft. We will let you know the following coming weeks. So the pattern of the exam is all written, and 25 to 30 questions you can expect. Out of 25 or 30 questions, 50% are multiple choice, and 50, the rest of them are open-ended questions. 1 or 2 is essay type questions. So this is the course outline for next 15 weeks. So do you have any questions on this? Yeah, you can unmute and you can say if you have any questions, or we can go on to the next. Okay, so if there is no any questions, then we can. So in general, like the microscope, usually, this course is meant to be for, teaching, the students, those who wants to, use for their studies, those who are working in the lab or PhD students. So we started this with 3 students, those who are interested to learn this. So some, like after the lecture, 1 or 2 weeks, we bring them student to the lab and ask them to learn the techniques and the process, how we are processing and everything. But nowadays, due to the COVID and other things, mostly it is remote and everything. So feel free to reach out to me if you have any interested in working, the lab, learning this. Always you are welcome. So those who already in the lab, as a lab rotation or research experience. So the safety in the lab is very, very important. So general basics is all the labs are same. So some of them, like specific places, where the instrument or the chemical, special chemicals, that is when you are going to use is, learn extra care. So always when you are working in the lab, use gloves and also wear lab coat. So that is not only protect your, yourself, but also, you should not spread, to your colleague or other lab, working along with you. Also, like always use the closed-toe shoes when you enter into the lab. Is a safety for your toes. If something fell, or if when you are working on the chemicals, acids or other things, if it is a spill, immediately that will, if your skin is exposed, that will be immediately affected. So always make sure that the standard protocol is. Already, the REHS, Rutgers Environmental Biomedical, Council is here. They will teach you. They give, online refreshing courses. The biosafety, how you are be in the lab. You have to get, certificate for that. So they teach you all how to be in the lab. So always, when chemicals means how you are going to handle, because you are not only just to use one chemical, but there are several chemicals, several acids, detergents, and so on. So we have to know exactly, how we can segregate the chemicals, because you cannot keep, one chemical and the solvents together in the one rack. That will be something fall or mixed, that will be giving different reactions, so that, handling. Similarly, the disposable, the chemicals, and also other, how you have to dispose. You cannot pour all the waste liquid into one bottle. So you have to know all these details when you attend this, orientation, the biosafety, orientations. Also the cleaning and exposure. So the cleaning is very, very important, because it is not only to, protect yourself or the instrument or the area of where you are working, especially in the bench or weighing or, hood. Wherever you are working on any chemicals, you have to make sure that clean them before you leave. Otherwise, if something spill, and if you are not cleaning these chemicals or solvents or salts, if you leave the places, if somebody else come there and if they work without knowing that there is a, chemical or salts or solvents, then he will be exposure to this and affected. So always, in the lab, it is not only to protect yourself, but also in the lab with the other colleagues and, students. So that is very, very important. This is not only for this, but whoever working on the lab, or in the future when you are going for a researcher or dentist or, doctor, this will be important. So here, dealing with the different chemicals and then different instrument. That's a light microscopy, fluorescence microscopy, laser capture, micro dissection, electron microscopes. All equipment is high, electricity consumption, so that we have to make sure that, you have to protect from this, accidental exposure or shock. So make sure that. Also, the laser capture micro dissection has laser. It is a mild, but still, we have to make sure that to protect yourself. So many chemicals, when you enter into the lab, you can see different chemicals. That's glutaraldehyde, paraformaldehyde. This is two important chemicals for microscopy. That's used for preservatives. So these are, when you exposed and you have a higher concentration, this will be a carcinogenic. It causes some people allergic and very sensitivity too. So make sure that we always use these chemicals in the, hood. Cacodylate salts are very toxic and carcinogenic. So which contains 50% approximately arsenic and readily absorbed through skin. So always make sure when we use this type of salts. So osmium tetroxide is another fixatives in the electron microscopy, is very toxic, and when it is irritant and volatile. For light microscopy, we use a propylene oxide, and this will be highly inflammable. Also, it's carcinogen. For electron microscope, you use embedding material to hold your samples is resin components, which is also carcinogenic. Some have allergic reactions when using, along with the antihistamines. So another chemicals commonly used for uranyl acetate lead citrate, in the electron microscope, that is also, toxic, and usually the uranyl acetate, we should dissolve this in ethanol or acetone, so that when it is spill on the skin, it is easily observed by the skin, so that that will be harmful to the humans or animals. So we have to be very careful with the handling of these chemicals. So alcohols, we use this as for solvents when we use dehydrates and cleanings purposes. We always mix the the powders, salts with those who does not dissolve in water. We use this. So liquid nitrogen is one of the dangerous, chemicals. This is a gaseous, and when it is, minus 180 degrees centigrade, and we will discuss about what is the purpose of liquid nitrogen in microscopy and how we can handle this. Physical damage. So when we use, razor blades and glass knives, these are all, important, and using frequently in the lab. So we have to make sure that careful with along with this. Finally, the disposal. That's most important thing. It's not only here at home or wherever you're working, the disposable, you have to appropriately dispose the, chemicals and other waste in appropriate containers. Always clean up any spills and messes after you done with your experiments. Nowadays, most of the laboratories stop using the mercury, but some old machines or some experiments still it is required. So if there is any spill or something, you have to call biosafety personnel. They will have a special kit and come and clean up. Always make sure that treat the cleaning material as a hazardous waste always. Any instrument, not only microscopy, any instrument, you have to clean up the equipment after your, when you use this. So this is the basics of, how you can prepare yourself in the lab when you come to work. This is the basics. And, if you are interested in the working in any labs for your, lab rotation or research, interest. So this is always you follow, these kind of guidelines, and also get orientation from the biosafety. They have refresher courses and course in person to teach you how to be in the lab to protect yourself and others. Okay. So this is, overall, our goal is next is we wanted to learn about, what is a microscope. So before you go into the details, how this microscope, evolved from a basics to advanced now with AI, artificial intelligence. So how these, all these centuries, so they developed, the microscopes. So we are not going to go in detail of each and every milestone, but very, very important milestone in these, years, how these microscopes, are developed until, now. So what is anyway, the microscope? What is microscope? Why do you want to use the microscope? As we have eyes and we can see all the materials, and why do we have to have the microscope? The microscope is an instrument for viewing objects that are too small to be seen easily by the naked eye. We can see the materials and objects and other things that bigger enough to see. If we cannot, see any materials, that means your eye power is, have some problems. So what we do is we go to ophthalmologist or optometry and check, and we can wear the, eyeglasses to magnify the images so that you will see the images clearly. So similarly, like the one which you cannot able to see in the naked eye, that means it is very, very small, that is micrometer, micro size, nanometer size, so that you cannot able to see through your eye, so that you need something to magnify the object. So that is we use called as a microscopy, the instrument for viewing objects that are too small to be seen easily by our eye. So that based on that, in 1590, two Dutch eyeglass makers, that is Janssen and Janssen, they experimented with multiple lenses placed in a tube, and both were forerunner of the compound microscope and the telescope. These guys, what they did is they have a multiple glasses, lenses placed into a tube, then they observed. So based on that, 1665, Robert Hooke, he looked at the silver of, cork through this microscope lenses and noticed some pores or cells in it. So after 1674, Leeuwenhoek, he's a business guy, built a simple microscope with only single lens microscopes, that is a glass beads in metal holder, to examine blood, yeast, insects, and many other tiny objects. So he only described the three shapes of the bacterial cells, first time. After that, 18th century, there are technical innovations which improved the microscope's lens, combining two types of glasses, which reduced the chromatic effect, the disturbing halos resulting from differences in refraction of light. So that is another milestone, that to reduce the chromatic effect, so that you will get a detailed and clear images. Based on that, in 1830, Jackson Lister, he reduces the chromatic effects using the several weak lenses together at certain distances. They use the lenses, different lenses, to place them in a particular, distance with each other, so that they develop here, clear, get detailed images with very clear, and that will reduce the blurring of the image. So this was the prototype for the compound microscope. That is more than one lens which you utilized in the microscope. We called it compound microscopes. So then 1872, Ernst Abbe, he derived a mathematical formula called Abbe's sign conditions, which provided a calculation that allowed for the maximum resolution in microscopes possible. So when you increase the images, using the lenses, the resolution is also important. Then only you will get clear images. So that's the one he developed, derived the formula how to increase the magnification without compromise the resolution. In 1903, Zsigmondy, he developed the ultra microscope that could study objects, below the wavelength of the light. So that is another important milestone in the 19th century. And for that discoveries, he got a Nobel Prize in chemistry during 1925. In 1932, Zernik, he invented the phase contrast microscope that allowed for the study of colorless and transparent biological materials. So for that, he got Nobel Prize in physics in 1953. 1931, Ernst Ruska, who co-invented the electron microscope. The electron microscope depends on electrons rather than light to view an object. That is another important milestone that, in the light microscope, we use the light source, as visible light. We have some limitation in light microscope. But if you use the electron microscope as a light source, then you can overcome the, problem whatever we are getting from the light microscope. So this is another important milestone, that is, even you can able to make it, observe as small as the diameter of an atom. We will discuss this in details later, how light microscope and electron microscope, advantage and disadvantage, everything. Then 1935. So Max Knoll, who is demonstrated the theory of scanning electron microscopes. So that is, a very important another milestone for that, the theory, and they developed the scanning electron microscope. So Knoll and Ruska in 1986 got Nobel Prize. Also 1938, first scanning electron microscope was produced by von Anden. During 1939, Ruska and von Boris both are working together and developed a first commercially available electron microscope. And then 1981, Binning and Rogerer, those who invented the scanning tunneling microscope, that is a three-dimensional images of the objects. And for that, They got a Nobel Prize in physics in 1986. So this is the basics of the theory and the milestone from how, yes, from a magnifying glass, and then from magnifying lenses and different compound microscope. That is one, instead of one lenses, they use two lenses, and then improve the chromatic, reduce the chromatic effect and increase the resolution, so that they developed different electron microscope. And these are all very, very important, and understand the history of these microscopes. So the next topic is light microscope. So what is the light microscope? Why important this? And what are all the different techniques available for the investigators, scientists, pathologists, and how they improve the quality of the samples preparation, observation, and interpreting the data. So we will discuss that next class. So the light microscope has become one of the most recognizable symbol of science. Even today, we have a lot of different sophisticated, high-tech microscopes is available, but still the light microscope plays important role in this science and technology. The optical microscope, often referred to as a light microscope, is a type of microscope which uses visible light and a system of lenses to magnify images of small molecules. That is the light microscope. The light microscope is called optical microscope. Sometimes they refer to as, the light microscope is a type of microscope which uses visible light. That is the light source we use for light microscope. So with that, we wanted to look how a human can view any objects, and how small objects he can able to see through his naked eye. And if you use light microscopy, how small samples you can observe under light microscope. If you cannot see that through this microscope, then what else you can look for? That is the electron microscope. So in this picture, when you look at this, the unaided eye, which you can go like a 1 millimeter size, you can able to see, or a little bit further with 50 mm. So you can able to see through your eye. If the smaller than this size, then you need to have light microscopy. So the light microscopy is, you can observe eukaryotic cells, nucleus, most bacteria you can see under the microscope. You can see mitochondria, also mycoplasma. Also you can try to see through light microscope. If you really wanted to see virus, proteins, lipids, small molecules, atoms, ribosomes, endoplasmic reticulum, nucleus. Now, if you wanted to see all these, means you cannot able to do it in light microscopy. So that you go for electron microscope. So that means like, now what kind of microscope we can see, and how small these molecules are, any samples. So the next thing is, usually you have microscopes. many people, your colleagues are using this microscope. So some samples, when they prepare, it require high intensity of the light source because they took a sections with the thick. That means you need to have, the bright more, so that only that will penetrate to the samples, the light, then you will get images. If it is a thin, the light source is, less, you require intensity. So people are, do this increase or decrease the light intensity depends on their sample. But when we go there and when you see your samples, you have to reset again all of them with your conditions. So that means after you use, you have to bring back that to default settings. So every time when you go working on any material, you have to bring that back to the default settings. Then only, the other people will come and work on the same instrument, it has to do for their own. So the student microscope we use, you can easily carry them from one place to another in the lab or lab to lab, so that, it is a small and you can easily carry that instrument. Not like we have now bigger microscopes with the sophisticated digitalized. It's not easy to move, but the student microscope still you can have. Always any microscope, when you're working on that, is at least you should turn off the lights and turn off the electric, connection, electricity, or unplugged, so that, if there is any power outage or any, things that will affect the microscopes. Also, most of the, good quality of microscopes, the light source, it is inbuilt within the microscope, so that we have to be very careful and, turn off these bulbs because it is very expensive. Always you have to clean the microscopes, especially on the stage and lenses, before putting away the microscope. The reason why you have to clean them is because we don't want to contaminate your sample to others, or if there is any dust for prolonged, if you are not using this. So there are always, clean up the area and close them in the cover and leave it up. Also, when you observe your samples, the focus is very, very important. So always you have to gently adjust the focus. So you have two different type of focus knobs in the light microscope. One is coarse, the other one is fine. So always you have to work first with the coarse, focus, so that you can, the objective lens between the distance between the objective lens and the stage, you bring back and set the right, distance to view clear images. How do you say that it is a good quality of microscope? When you go in the lab, how can you say, oh, looks like a very good microscopes? But you have to tell the reasons why it is good quality of microscopes. So one of them is the built-in illuminator. The light sources should be attached permanently into the microscope, so that you no need to worry about that, or every time if it is it's not connected to, not inbuilt with the microscope, then there are a lot of problem to the intensity of the light source. Every time when you go back and forth, that's different. Also, the adjustable condenser with aperture diaphragm, that is important to get a good contrast. Also, which gives you control the mechanical stage. That is another important aspects of good quality of microscopes. Also, the binocular eyepiece tube, and also we called it as ocular lens. That is also important, and these are all the good quality of microscopes features. Also, how to improve the light source, the intensity of the light, and the path of the light to your samples. So for that, a simple adjustment on condenser lens aperture. So this technique developed by Collar scientist. So after his names, we called it as a Collar illumination. Also, how we are going to get the images. We're going to discuss that later. What are all the different techniques in the bright field microscope? Light microscope is dark field microscope and phase contrast microscopes. That these two important microscopes is, techniques will give you a lot of information of your samples. So this is the basics of your light microscopes. So as you know that, this is ocular lens, or we can call it as eyepiece. So this is, the two is objective turret, that is to hold the objective lenses. So the each objective lenses, this one, the three, has a different magnification, that is as maybe 10x or 50x or 40x, means 10 times magnify or 40 times magnify. Or this is a lens. Based on that, you will see the images. So this is the 6 is stage. So here, this is to hold your sample, the slides. You can place the slides here with your samples. And this is a 9 is, the back and forth, you can do the jack with the stage to adjust, to bring the samples parallel to the objective lens. And 8 is, diaphragm and the condenser. So that will, you can use this for color illumination settings, that is to improve the light source. And then, the 4 is the coarse image, like a focus. And 5 is a fine focus. So this is the typical, so the light source is underneath, it is inbuilt in the microscope, so that, whenever you turn on the light source, you can see the bright light source. So once you turn the light on, it will be passed through the condenser lens and, go through the stage, and here your sample on the slide, and then the objective lens through, you can see in the eyepiece. So this is a typical, good quality of microscope to use in the lab. So there are different type of microscope. Is one is one with the lens, one lens, a simple microscope. We call it as a one lens to magnification. That is a simple microscope. Another one is a compound microscope, that is more than one lenses you can use, that is objective lens. We call it as a compound microscopes. So there is another, 100x microscopes with oil immersion that is another, microscope lenses that is also attached to this, microscopes. So the basic components of a light microscope is, as we mentioned, this is eyepiece or ocular lenses, objective lenses, then the stage and illumination sources. The eyepiece or ocular lens is a cylinder containing two lenses to bring the image to focus for the eye. For example, when we have look at the microscope, the slides with your samples, when I go and look at them, my eyesight is different than your eyesight. So what I can do is I can adjust the eyepiece or ocular lenses, to focus to my eye. Then after I observed this, then if someone else come and check the same, they have to adjust the ocular lens or the focus to, to their eyesight. So for that, the eyepiece or ocular lenses plays important role, that is to bring the image to focus for the eye. The next is objective lenses, that is it projects the magnified image to a fixed position behind the lenses. Each of them is a different magnification lenses available in the market. You can select which one you want to utilize. The next is a stage, that is the platform below the objective, which supports the specimen being viewed, is stage. The illumination source, that is the light source. Below the stage, the light is provided and controlled in a variety of ways. So that is another important, component that is attached to the microscope. So this is a simple basic microscope, has only one lens, and you can see two lenses, that is eyepieces and objective lenses. And this is the stereo microscope is, you can use to the samples, which you have a standard, magnification. And if you wanted to see a bigger samples on the surface of how it looks and everything, you can use this stereo microscope. So this is the basic microscope, the compound microscope and the stereo microscope. So we will discuss this, how important this compound microscope and the stereo microscope, we can compare these two with the scanning electron microscope, and this one is for transmission electron microscope later. So with this, I think today, first day, and I will stop now. We can discuss the next class. So next class means the following weeks I won't be, call for the class, and maybe I will forward the lectures. So for any questions, do you want to ask me before we can leave?